A very good morning to our resource person, Dr. Sanjay Pai, and dear delegates for the QIP. We start with this morning session with a brief introduction of our resource person, Dr. Sanjay Pai, who is presently a professor in pharmaceutical chemistry at Government College of Pharmacy, Goa. He has graduated from Bangalore University and also is a postgraduate from the same university. He's been awarded for a PhD on his thesis titled Drug Design in the year 2003 by Bangalore University. He specializes in the area of synthetic chemistry and pharmaceutical analysis with special taste and research in the domain of impurity profiling, analytical method development, and drug stability studies. He's been in academics for over 30 years and he carries credentials of teaching courses in pharmaceutical organic chemistry, medicinal chemistry, and pharmaceutical analysis to thousands of knowledge seekers. In research, he has guided more than 100 students for their postgraduate in pharmacy and successfully guided 3 PhD students. His active involvement in research guidance has brought many accolades with more than 100 publications in peer-reviewed national and international journals and scores of presentations and conferences. He loves to share his knowledge on recent developments in his area of interest on regular basis through lively presentations conducted by QIP centers across the country. He has been on the board of studies and examination of several universities. He led a team of vibrant professionals from academia and industry as editor-in-chief during 2007 to 2013 in pursuit to restructure one of the oldest popular journal, Indian Journal of Pharmaceutical Education and Research, referred to as IJP-ER. Efforts turned fruitful as the journal secured impact factors for the first time during that period. He has contributed immensely for professional development activities of APTI for over a decade. He has acknowledged invitations from PCI, AICTE, AQIS, NTA periodically for many of their confidential and routine activities for over a decade now. And here also I would acknowledge his association with the Cayley Institute for over a long time. With this brief introduction, I welcome you, sir, on behalf of Cayley College of Pharmacy for this session. Now you can take over the session, sir.
Madam, thank you very much. Good morning to all of you. Good morning to...
You're not audible clearly, sir.
I think we have about 20 odd participants. Just let me know if I'm fine now. Am I audible?
You are, sir, a bit better audible. Yeah.
Okay. So I welcome one and all the participants who are with the QIP program. I think we are towards the fag end of the session, and I got an opportunity, some of the routine incidences, or I can say the routine difficulties or challenges that we face in our routine professional activities. I'm not very sure how many of you are in the domain of the analytical method validation group as such among the participants. But it is good to know that since most of the participants are teachers of the various academic institutions who may already be into research or would have come to the current position on the basis of their research accomplishments, perhaps it's an opportunity for them as well to introspect, and then perhaps uh, I would put it this way, that they can perhaps design the future of research in such a way that whatever deficiencies or whatever merits they have experienced so far in their research arena, perhaps if they find some deficiencies in their research arena, they can overcome through these shared experiences. And today I took a topic which is hotly debated these days. How good are our testing systems? We are talking of quality, we are talking of quality enhancements, we are talking with regard to fall in the quality of products that are being dumped into the market, etc., etc. Now, all this is happening, as you all agree, because our testing systems, there is a big question mark put on our testing systems itself these days. To what extent our testing systems are performing in order to ensure that quality products are pumped into the market? Quality systems are available, but what are the forces which are preventing perhaps the system to adore and then ensure that we follow the best of the testing systems in order to ensure that the quality is inbuilt into the products? So let me just share some experiences. As I said, basically all of us have got different experiences. The experiences which I have faced as challenges, based on my experiences as a researcher, my experiences as an evaluator, my experiences as a team leader, my experiences as a group member. Some groups I am just a member, and whatever facts which I have collected, perhaps I consider them as the challenges. And as you all agree, if you are an analytical chemist, you are in that group which perhaps many of you are hated the most in your organization. The sincere you are, the hate factor is the maximum. The reason is, when you are an analyst, you are required to identify the strengths, the weakness, the opportunities, and the threats for your system. Whether it is in analysis, whether it is in some arena of your interest, well, people will always tackle you or look at you in a different frame of mind, because you are the one who will identify the weakness in your system. Well, you are the analyst, and perhaps you should find a way, in such a way that the quality systems are upheld, there is no compromise on any ethics, and that will ultimately lead to the best possible testing system available today, not only to develop the quality of the personnel, but also to develop the quality of the products that are produced in the industry. Well, last 2 years, there's a lot of talk about a molecule ranitidine, which was sold in tons every day, and suddenly a laboratory in United States of America, they said that, when we have analyzed ranitidine products sold in the market in the US, we found abnormal amount of NDMA, N-nitrosodimethylamine. This N-nitrosodimethylamine is a carcinogenic agent, and then its present raised eyebrows, and testing of this ranitidine products giving abnormally high amounts of NMDA really created some kind of flutter around the world, especially the companies targeted were the ones which were selling ranitidine in big volumes, yeah, including the multinational companies like the Glaxo had an issue with regard to this ranitidine. I remember the articles describing, millions of tablets which were withdrawn from the market once this matter started dampening the prospects of those industries. Then later on, FDA came into picture and they said, "Array, there is no issue with regard to ranitidine. It is the problem which is associated with the testing methodology, which which gave an insight that the NDMA which was found in abnormally high quantities was on account of a defective testing method, and that testing method, the GC method, we all know, that was responsible for release of this NDMA in large amounts. And perhaps even if ranitidine was not containing NDMA in the formulation, but the process that was followed, the GC method that was followed, released a lot of NDMA during the testing procedure, which perhaps was the reason for abnormally high quantities of NMDA present in the product. So the US FDA at that time in 2019, when this issue had was blown out of proportions that time, they gave a clarification, and they even insisted that why don't you take up a LCHRMS method rather than a GC MS method, because it was the GC where the temperatures were high, and at high temperatures there was release of NDMA, and this NDMA, since it was present in large quantities and had issues with regard to its carcinogenic activity, which ensured that the GC method was withdrawn. And from then the US FDA, which insisted that the LCHRMS method be replaced, let it replace the GC method, brought some kind of sanity in the world market when it came to selling of the most popular drug, which is ranitidine. So what I would like to say is the testing methods, which most of us consider as non-priority part of research, is something which has assumed a form wherein, unless the testing methods what are evolved are good enough, you cannot expect the verdicts which are given across the purity and impurity of the drugs to be acceptable. So it's the testing methods which will decide how well the products will be accepted in the market. Well, many of the methods which are proposed these days, which are the pharmacopoeia prescribes use of these kind of methods, they have some kind of issues when it comes to drugs being analyzed, which are stored for longer durations. So basically, you ensure that the methods that are taken up are robust. The robustness of the methods is something which is the something which people are on the lookout for these days, to ensure that the testing methods are satisfactory and good enough, so that the products, you don't have to compromise with the quality of the products. Let's have a look at some of the products which have faced some issues, and that has led to many big multinationals withdrawing or recalling those products. I'm just going to read a few of them here. You find thousands of examples over last decade. Even Hospira, had to withdraw metaxantrone injection because of the failed stability specifications. Apotex piperacillin tazobactam injection, since they found high level of impurities. Remember, these were the products which were already there in the market, and the companies had to initiate a recall simply for the fact that the testing methods that were applied for these products were not robust enough to predict the formation of the impurities, which are basically degradants. And then, when some small laboratories used certain methods in order to identify these impurities, that brought in alarm to these big MNCs, and they had to resort to recall of the products. Most of the examples, what you find, I'm not going to read all of them, you will find that basically these are the degradants which forced these big companies in order to withdraw or recall the major brands from the market, simply for the fact that when these left the industry, everything was fine. But when somebody pointed out the presence of some degradant or some undesirable component, then these companies had to resort to and acknowledge, since their testing methods were not good enough to predict the recognition or predict the detection of these impurities. So what I would like to reiterate here is it is the testing methodologies which we use for approving the products, which fail because of the lack of foresightedness or lack of ignorance, or perhaps lack of those intimate steps which otherwise would have helped in prediction and detection of these undesirable impurities continuing to exist in the formulations. Let me just show you a few more examples wherein certain degradation products which are likely to pose some kind of undesirable effects have led to some kind of vigorous screening of the active pharmaceutical ingredients, and the limits within which these degradation products which are required to be present in these drugs have been found to have been declared to be abysmally low now, since they produce very lethal and undesirable side effects. Uh, so isoniazid, pethidine, lidocaine, gabapentin, furosemide, proguanil, rofecoxib, to name a few. And I have listed a few products which are the degradation products coming from these compounds. The list is very long. I just picked up a few popular ones just to give you a fair idea about what should be kept in mind, what should be borne in mind when you go for, devising a method which should also help in predicting as well as identifying the possible degradants which can form out of the products, so that the companies or the laboratories, when they are launching the products, they know that their products will not fall through somewhere in between, and days will not come when they are required to recall the products and face that embarrassment. So gone are the days when you would look at the product with naked eyes. Today, people hold lenses. Even after the product has moved out of the laboratory, people are after the products, trying to look at them with the lenses of the highest possible power in order to perhaps identify such undesirable products and make big issues out of them. Now, what are the challenges? Let me look at the challenges from the analyst's point of view. See, all these years, we have been very comfortable developing methods. We say that analysis is the most simplest branch when it comes to MPharm, and we have reached a stage wherein we have even decided that analysis need not be given an independent identity, since it is a property of everybody. Analysis is required for a pharmaceutics technologist, pharmaceutical technologist, it is required for a pharmaceutical chemist, it is required for pharmacologist, it is required for a biotechnologist, it is required for microbiologist. So it should not have an independent identity, but maybe it can have identity in such a way that all the technocrats or all the technologists are equally responsible for the whatever developments happen in the area of pharmaceutical analysis. Everybody is made equally responsible for it. So gone are the days when we used to train our students how to sail, right? How to sail in the river. So that is what we used to do all these years. We ensured that students learn sailing on, on waters where currents aren't there. Now the situation has come wherein you need to train your students in such a way that they should be able to sail in seas and oceans where high tides are common features. You need a lot of mental and physical effort if you have to do it, and this also will therefore test the ability of the teachers who are guiding such students these days, in order to assist, in order to train the students in such a way that the students develop the skill, develop the the qualities which are required for developing new methods in such a way that the products don't fail. This leads to some kind of a hypothesis, as I would put it, because when Charles Darwin said, survival of the fittest, those days, uh, he said that it is not the strongest of the species that survives, nor the most intelligent, but the one most responsive to changes. That appears quite true these days, not from the point of something which has life, but something which does not have life too. Now look at the performance of the analysts over a period of time. The analysts are struggling since they are not responsive to changes. You look at the our curriculum when it comes to BPharm. You look at the pharmacopoeial standards today. Now, what perhaps was the training component maybe 25 years ago, we have not changed anything from there. Look at the kind of technology that has got into the pharmacopoeial monographs. Unfortunately, we have refrained from transforming ourselves from what was the technology in perhaps 1985, 1990 to 2020. What is required in 2020? Now, how many experiments, how many monographs insist that you require an HPLC to perform an assay? And how many experiments actually we do in BPharm? We say that our BPharm is meant for a student to work in the industry. Now, we ourselves, the system itself, unfortunately, we are not responsive to change. The system itself is not responsive to change. And that question once again lingers in the mind. Uh, are our systems of education the fittest enough to enable the trained students to face the future? I think all of us as teachers should think about it very, very seriously. Now, where are the faulty methods? Where are the faults lying? If you know where the faults are lying, perhaps you will be able to identify the challenges and work upon them in such a way that you can try and paint those, or paint those, faults, and then see to it that they are plugged, and you can develop better methods which will have some kind of meaning, which will have meaningful outcomes when it comes to deciding the quality of products is concerned. So when you audit the findings where the methods are faulty, you will see this. I'm talking with regard to the analytical development. I'm restricting my talk only to analysis. You see the problem, the major problem associated. Now, this is my experience with regard to evaluation of thesis from the last, I'll take almost 10 methods. Most of the, the work that has been proposed in PhD thesis has remained incomplete because the validation methods which have been followed, they have not gone totally to completion. You look at the ICH norms, you look at what is required to be done, just look at what has been done, you will find a lot of gaps. Now, why those gaps are there? Because either to say that my method is not validated completely. Now, why it is not validated completely? Because my results are not conforming to the accepted norm. Relate my values and put them in the thesis. At the same time, I don't want to say that my method cannot be used. I always want to say that my method is the best. My method is no short and equivalent to the pharmacopoeial method. See, I want to finally summarize my observation that way. So what I will do is I will either eliminate some parameters because they are not fitting within the required range, or what I'll do is I will simply manipulate and put it that way that my method is as equivalent to the pharmacopoeial method, and as a result, I deserve to get a PhD. So that is my novelty. The other problem which is associated with the audit findings is that, I'm not able to provide adequate information. I have found that, many a times the calculations which are done, they are not making sense. The calculation is done in order to ensure that the values what finally the analyst is doing should conform to the acceptance criteria. So there is inadequate method validation, which unfortunately doesn't provide complete information, which otherwise would have, declared that the method is faulty. Lack of appropriate controls could be probably, underlined here. You don't there could be determinate or there could be indeterminate controls, something which can be under your control or some things which cannot be under your control, which ultimately affect the integrity of the validation. So the validation protocols, what is followed by the analyst, is a big question mark, and that could be one of the major reasons for development of these faulty methods. Now you just go on listing out why these methods, what have been developed by the analyst, are faulty. You may not declare it openly, but that will certainly pass through your mind. It could be lack of foresightedness. I just want a PhD. I don't want my methods to be applied anywhere. So I don't declare all the information. So I will just say that, I have done something which has given me this result and I deserve to get a PhD. So my objective is to get a thesis which cannot be challenged, because I'm not giving you that data which will be controversial. If I give you that data which is controversial, then perhaps, those gaps will be identified by the evaluator, and I may not be able to justify, my entitlement for the PhD. That is the major reason: lack of foresightedness, insincerity. Now the methods become faulty because I would like to ensure that the values what I am getting fits within the accepted range as declared by the pharmacopoeia or the ICH. The lack of commitment. See, I'm doing this only because I want to complete my work and see that I submit my thesis. There's lack of commitment. Self-centered. Anyway, the data what I am presenting will give me a paper, will give me a PhD, and I'm not bothered about how this method is going to be used by the industry. So a very self-centered approach. Static. This is what I have learned in B Farm. This is what I have learned in M Farm. I don't want to grow beyond that. I don't want to present something which is novel. I think this is novel because I don't study. I don't refer to journals. And whatever I know is probably something which no other person knows. That is the kind of static mindset which many of us have. Contentment. Okay, I can do this and this will lead to a paper. So I don't want to go beyond that. Now, this kind of contentment also creates problems when it comes to which will ultimately lead to methods that are faulty. Or ignorance. This is the biggest issue. Ignorance. I don't refer to what is happening around the world these days. Whatever I know is the ultimate. Nobody can challenge me. I don't know what is happening around me. I am too ignorant. This is what I have learned the best in my days, and I would like to still continue with it. That is called ignorance. And this is what is happening for non-academicians. Many of them, unfortunately, are in the regulatory positions. Now, where it hurts, these kind of problems associated with the faulty development of methods, where it is actually hurting. If you really look at that, then I think the answer lies once again in that, the potential of commercializing the product is getting hurt because, companies are required to resort to withdraw the products which are already there in the market, or there may be considerable delay in starting certain clinical trials, or maybe shipment of the product. Considerable delays can happen. Maybe the incomplete method validation can impact the stability studies, and that is where the issue is. I have showed you some examples wherein in the course of time some formulations are giving up some impurities, and they were not predicted because of the lack of foresightedness by the analyst, and as a result the product is already out, and somebody else, some laboratory somewhere in the corner of the world has raised a big question mark because they have developed a method and they have applied it to the formulation available in the market, and then they have identified that impurity. The case of not exactly, I will say, ranitidine, because ranitidine had a different issue. The testing methodology itself had a question mark, and the methodology, the problem associated with methodology gave an insight into the presence of abnormally large quantities of that undesirable impurity. So eventually what I would like to say after having last 20 minutes, something which is ensuring that our methods are result, we should be thinking about we should be planning or designing our methods in such a way that whatever is likely to happen in the next 3 years or until the shelf life of the product is that the method should be robust enough till at least that product is able to be sold in the market. So the lack of robustness is the major issue, and that throws a big challenge to the analyst. Now, what is this robust method development? Now, it's something which we are all aware of. Unfortunately, the theoretical aspect is something which we are aware of. When it comes to the practical part, we don't accept it. That is the biggest issue. See, when we say, please write an analytical method, what is the analytical method that you have followed? When people come for an interview, when you ask them, they will say that, take the drug, dissolve it in a suitable solvent. Now, which is that suitable solvent? What volume is required to dissolve that substance? All these things are considered as minor variables. But when the method is robust, everything has to be crystal clear. So detailed steps are to be, written down. And when you write detailed steps, anybody in this world should be able to follow them word wise, and they should be able to reproduce the technique. Unfortunately, what happens, since we ourselves are not confident that our method is going to work, if I do the same experiment tomorrow, what generally an analyst does is leave long gaps in his procedure so that there is a way out tomorrow. If the method is not properly working, the analyst will say perhaps somebody who has reproduced his method has skipped some steps or has not done in the way as it is required to be done. Gentlemen, when that step, the detailed steps itself are missing in the procedure, then how can you expect somebody else to reproduce what you have done? So the major issue is writing procedures. The procedures, when they are written, they are so defective and they have gaps that something which is required to be practiced is generally omitted, and that could be one of the reasons for deviation of results when the same experiment is done in a different laboratory by a different analyst. The second part, the most important part, is the way the samples are prepared. When you write the procedures, how the samples are prepared, that's the most important part. The method of mixing, the method of making a solution, method of mixing, sonication, what is the frequency used, what was the temperature when the mixing was done. We ignore small aspects, small points, and later on make a big cry when those methods don't work or are not reproducible when you go for inter-day experiments. Many a times the standards what we use, they are not good. The reagents, what we adopt, the reagents, what we use, we don't verify the strengths of those reagents. One good example is hydrogen peroxide. In the stress degradation studies, we are using hydrogen peroxide to check the ability of the drug to undergo oxidation, right? Oxidative stress. And how many times we analyze that hydrogen peroxide to ensure that the hydrogen peroxide is of the required strength? We never do that. We assume that hydrogen peroxide is of the required strength because it has been opened from a new bottle. And we know what is the stability concern of hydrogen peroxide. Many a times we use HCl. I see my students using HCl and sodium hydroxide when they are performing stress degradation study, especially under that forced stress with HCl and sodium hydroxide. I have asked many students many times, how many times you have assayed that HCl and assayed that a sodium hydroxide because it is written 0.1 normal. How many times you have assayed during the course of your project? Forget about the person giving an answer. The person could not even satisfactorily answer the standard methodology used for assaying sodium hydroxide and hydrochloric acid. The standards, what I mean to say, the standards that are used, they are not strictly adhered to. And this again leads to a method which is not robust. Reference standards which are used are generally the gift samples. How many of us do monograph or analysis of those samples which come from the industry? We always assume that what comes from the industry is Amrut, right? It is the Amrut. And we think that it is 99.9999% pure. You should know that many of these samples which come as gift samples, they are samples which were sampled from the warehouse for testing, and then they are perhaps if you have come across some industry visits, you must have seen that these samples which are on the weighing pans, when they swipe the dirt from that, they all collect it in one packet, and then they just dispose that packet as the gift samples. So what other sample, in case if it is present on the balance, will become part of your gift sample? So it is our duty that when we receive these gift samples, we perform monograph analysis and ensure that the samples are pure enough. Many a times I have seen that what we receive as gift samples are not to be considered as reference standards. Some industries, they put that statement because they don't want to receive the compliments, the adverse compliments later on. They say that this reference sample which is sent to you, or this gift sample which is sent to you, shall not be deemed to be a reference standard. It is very clearly mentioned. And many a times till now, we always have made it a habit to use these gift samples as the reference standards. The apparatus what we use can become a problem. Many a times the apparatus what is sold, they are not the volumetric apparatus. In fact, how many of our students know what are the volumetric apparatus? During the examinations, whenever I have asked which are the volumetric apparatus which you have used, the first answer that comes is a measuring jar. I'm just leaving some thoughts to mind of our teachers. The measures that are required to be undertaken by our fellow teachers in order to groom our future analysts is something which I think we need to have a great concern on that. Many of our students know only some formulas for calculation. See, we would have taken some representative experiments and we do not show them what is the origin of that formula. Because somebody in some other institution is using this formula, I am using this formula. Now there is a source for every formula. Either the factors, the dilution factors are different, or the pharmacopoeial factor is different. Many of the students are not aware of that. They simply download or borrow some material from their friends from some other institution and will be using that same formula, and they assume that that formula is common for getting the final answer. Or I have seen some students adopting and designing some formulas in such a way that the answer comes between 99 to 100%. So this is another issue which is of grave concern when it comes to the robust method development. Here just doesn't mean the technology behind the method that is adopted, but every step that goes into assay of a particular substance or estimation of a particular substance comes under that broad term robustness. Ultimately, the procedures will be too complex. When the you should not worry if the finally the procedure that you evolve after ensuring that all robustness study is in place, should not worry that no, my procedure will become too complex, too extended, too expensive. These are the terms which I don't find somebody summarizing at the end of their paper or at the end of the thesis. How many of you must have seen, a thesis ending with these statements? My method has become complex. My method has become extended. My method has become expensive. Generally, you read any paper or any thesis, ultimately it ends with one summary. This method is simple, less time consuming, and cheap. 97% of the thesis ends with these statements or synonyms of these words. The procedure that was developed was simple, less time consuming, and cheap, or less cost. It's less expensive, something like that, which is exactly the opposite of these words which I say. Procedures have become more complex, become extended, and become expensive. Remember, gentlemen and ladies, when your method becomes more complex, it becomes more robust. When it becomes extended, it requires extraction step, it requires additional steps, then your method is likely to be considered more robust. When I say robust, your product is unlikely to fail, right, once it moves out of the industry, and will remain, because so many steps are already inbuilt that you don't have to worry about something that is somebody complaining that something else is present in this compound which has not been reported at all. So that issue doesn't come. Don't think that anything that is less cheap is very good. No, there may be very less instances like that. Many a times when you add steps, when you add more reagents, when you use high-fi equipments, high-tech equipments, the cost is definitely going to be more. So whenever I find a thesis using these words, method is simple, less time consuming, very cheap, I look at that thesis with a lot of suspicion, because those methods, eventually you can prove that the methods are not robust. Now, why these analytical testing procedures are important? Without testing procedure, without testing, a person cannot be declared as COVID positive. Can a person be declared COVID positive without testing? Like that, without testing, you cannot declare the quality of the product. So testing is the most important component of a product, without which the product itself cannot be considered fit enough for consumption. Agreed? Without testing, you cannot allow a person to complete his degree and claim that he has come out of a particular, he has got a degree in flying colors. I'm just trying to compare so that you understand the importance of testing. What has happened today is, whenever testing becomes a talk in the department, it is given less emphasis. Array, what is there in analysis? You develop some method, you prove that my product is passing. That means you are compromising. You are compromising the ethics, isn't it? I'll come to that particular part. So analytical testing procedures play a very critical role in equivalence and risk assessment and management. So if you don't have a good testing procedure, everything is getting compromised, right? It helps in establishment of product specific acceptance criteria. That, of course, we have to accept it. It ascertains stability. Now, this is another issue. If your molecule is unstable and you don't have a method which can identify the degradants which are formed in the industry, or maybe later on when it is in the market, then you're not very sure that the testing method that was adopted was good enough to predict the degradants which are formed out of this instability concerns. Now today we have stopped blaming on this personal bias. Earlier days we used to say that a testing person can be biased in the sense, I want this to be passed. I can simply say pass. There's no transparency. But today we are blessed with the technology which will ensure that the personal bias can be eliminated if required. If required, I'm putting it again uh, as with an underline there. But it's easy to get away with the personal bias because the most advanced technologies today have ensured that personal bias can be eliminated if required in a very easy manner, and consistency of results can be expected. Now, when these analytical methods are required, now there may be delegates from different branches. And many of you may be thinking, I am talking, I'm making this presentation from only the point of a pharmaceutical analyst. Gentlemen and ladies, no, I am talking the need for analytical methodologies, which is required for all of us. Whether you are in pharmaceutics, whether you are in pharmaceutical chemistry, whether you are in pharmacognosy, whether you are in pharmacology. In fact, today pharmacologists and pharmacognosy requires diverse types of analytical methods rather than a pharmaceutics person. So I've just listed out who all may be requiring this. And because there was a time when many students used to say, I don't want to study pharmaceutical analysis, because that is a branch where I'm required to use lot of technology, lot of work is there, and lot of volume need to be, lot of data needs to be generated, computed, and then it's hard work. But today, if you really recall what has happened during your research program, you would have spent 50% of your time doing only analysis. And perhaps sometime in your life, you must have thought, if I had studied analysis well, probably half of my problems, what I am facing now, because I have to depend on somebody else to get my results, would have been solved. This happens when many people, many researchers, they come to me for interpretation of this mass and NMR data. They come to me, they said, if I thought that, so much of intelligence is required for these interpretations, perhaps I would have studied these chapters very well those days so that I could have been self-dependent. So you will find your area of interest in one of these 8 domains, as I would put it. Either if you may be having the perhaps you may be in pharmaceutics or in cognosy or in pharmacology or chemistry. You will certainly find your domain in this list of 8. So going into the core of this analytical method development, the major objectives is to develop a test, to develop methodologies in such a way that you test a defined characteristic of a substance and ensure that it meets the established acceptance criteria for a particular characteristic. So your methodology should be such that you develop a particular technology to test a particular characteristic and ensure that it meets the established acceptance criteria for that. So whenever you do analytical method designing, all these parameters come into mind: specificity, linearity, limit of detection, quantitation limits, range, accuracy, precision. I'm not going to talk anything about these things, nothing in detail, but I will simply tell you, uh, inform you that whenever you get a thesis for evaluation, if you are getting it now, fine. Otherwise, if you get it later on, you please have a look at what has been covered in these parts in the thesis. Or if you have access to your students, just have a look at what exactly is happening when they are working on these parameters, when they are validating these parameters, you will always find that they will be looking for some shortcuts, some shortcuts so that I can complete this within a short duration of time. The shortcut could be either something which I have already listed. Take some glassware which is available, just follow the process blindly as it is given in ICH. Just follow the procedure, or take a look at some senior's thesis. Just follow the methodology routinely, and finally ensure that you get a value either with that manipulated formula or a formula used by somebody which has no relationship to the formula which is required to be used and which is required to be designed through a logic. Nobody applies that. Simply, I'll say nobody. I don't say nobody. Most of them don't do it. They simply ensure that you finally get an answer which will fit within the acceptance criteria defined by ICH. Now this is the major cause for that faulty method to be developed. The method is good enough to get a degree, but when it comes to application elsewhere, maybe in the same institution in a different laboratory, it doesn't work. Method developed in pharmaceutics department doesn't work when the same method is taken up in pharmaceutical chemistry department. Now, these are the four components that are to be borne in mind if you have to ensure that the data quality meets expectation. If the data quality has to meet the expectation and in such a way that tomorrow the method could be robust, then these are the four components that an analyst has to ensure that he has worked seriously. One is qualifying the instrument itself. The best analyst always suspects the instrument, whether the instrument can perform and give me the results as it is supposed to give. So the qualification of the analytical instrument becomes very important. How many of us ensure that the instruments are calibrated on routine manner? Unfortunately, what is required for calibration only is missing in many of our laboratories. How many of us have seriously thinking all laboratories have UV spectrophotometers, but how many of us have holmium perchlorate solution, which is an important component for calibrating our equipment? I think we should touch our hearts and see that, do something in a compassionate manner, train our students with regard to this particular aspect. And then, unless we provide a good stage for a person to dance, we cannot expect a dancer to perform well. The second part is to train the student the importance of what validation is, to be frank enough to deliver the results what he has got and not manipulate them, and then bring it in the range which fits within the acceptance criteria. The system suitability tests need to be done, whether the system what I am using is giving me agreeable values with regard to my tests done on a particular drug. The system suitability is something which we hardly or most of the time ignore, because the instrument itself calculates some variables like tailing factor, like height equivalent to theoretical plate. We always ignore, and we say that the system suitability passes and I should not expect any problems when I go for my validation. But there are certain, if you when you scrutinize these values many a times, you will find that the system is not fully suitable. The mobile phase, stationary phase, fitment is not fully suitable to give you a most robust method. With experience, perhaps you will be able to note from the data that is generated by the equipment that either the tailing factor has some issues or the theoretical plate has some issues. Consistency becomes the biggest issue when there is variation in these system suitability tests. The quality control checks need to be done periodically by the guides in order to ensure that the students are providing values which are consistently good and not resorting to manipulation. So analytical procedures development are primarily based on combination of mechanistic understanding of the basic methodology and prior experiences. This is where the contribution of the guide matters. In many institutions, the students are left on their own. They are developing something. They do not know what they have studied in theory, how it can be applied to what is being done in the practicals. If the guide's involvement is not present, then there is compromise on this particular aspect. There's no mechanistic understanding of the basic methodology. Just because senior has used a buffer, I am using the buffer. Arey Baba, what is the need to use that buffer of what pH? No idea. Now, this mechanistic understanding has to be imparted in the student if the student has to understand the importance of designing the method and prior experiences. This is important because only the guide has prior experience of handling these kind of issues, and you cannot expect the student who is touching the instrument for the first time, who is handling the instrument for the first time, to have dreamed of experiences. There's no chance of that. The experimental data from early procedures can be used to guide further development. The performance of the methods that have been developed need to be always evaluated, because when you evaluate the performance of the method, you will get an idea whether the method in future is going to work in the same manner as it has worked previously. I think what I'm trying to hint at is something wherein you will find that the method is not consistent. Each day the result is slightly varying, but I get a result which is very good today, which finds acceptance in my thesis. So what I got today, I will collect it and put it, right? So that is not the criterion. You should consistently evolve a methodology. The methodology should be such that any number of times the assay is carried out, the result should be consistent, reproducible, and it should say whatever it is. And many a times the method's performance can be judged on the basis of results that are obtained in the various steps of the finished dosage manufacture. In some stages, you will find that there is significant deviation. And when there is significant deviation, it only means that there is a lot of interference which is caused by either the excipients or the environmental factors, which once again is most likely to end up in a method that you will declare which will be non-robust. Now, who can do this method development? As I said, all of us require to develop methods. It's not the domain of a pharmaceutical analyst alone. When you're developing a new product, whether it is herbal or the synthetic, you need to develop methods yourself. You have to depend on this part yourself, because you know what has gone into making of that formulation. So you should be qualified enough. You should have studied pharmaceutical analysis in your bachelor's. You should have practiced. You should have got your experiences from various experiments that you have done. So you should be a qualified analyst, needs to be skillful. Observers are not very good in skills. Many of us, we are very good observers, but when it comes to skill, because we have not practiced it in the laboratories, we are less skillful. So a good guide is one who will wear an apron, stand in the laboratory with the student, and will experience, will share the experience along with the student in the laboratory, and then develop that skill, which most of us are missing. Expertise in sample selection is very important. How you prepare your sample, what solvent you will use, what pH is required, all this requires some degree of intelligence. So expertise is required. And most important is integrity, which is missing in most of the analysts, unfortunately. How? Now, how to develop methods? Now, who will develop methods? Who is the right Human, human resource who can develop methods is something which I listed. Now I will list how to develop methods. Ensure that the instruments that are being used are calibrated. The tools which are used are good enough. You have lots of paper available so that you document what you are doing, because nothing will stay in the memory for a long time. So the person should develop that art of documentation in writing down, in noting down whatever is being done, whatever is being practiced. Most importantly, use reliable reference standards. I've already highlighted on this. And the change control. You should be able to document whatever changes have been done on periodic basis and have controls in such a manner that if it is required to be repeated or reproduced later on, the same needs to be adhered to. The motto is very important. The analyst should always follow a motto. He should have the ability to provide accurate, reliable, and consistent data. That should be the motive of the analytical chemist. Unless the person decides that he will practice professional ethics, the top line, the top statement is not applicable. So if I have to provide accurate, reliable, and consistent data, whatever I get, if I have to present that, I should be bold enough and I should be practicing ethics. I know what is required to be practiced by an analyst, so I will practice the same. So give and take, that compromise should never be in the frame of mind of an analyst. The day that compromise comes into picture, the robustness is lost. The life cycle of analytical method. This is a very, very painful process. Many a times you see that you are required to recycle. Now, all of us, whoever have worked in the analytical arena, they are aware of all these steps. But many a times we only engage in steps where we don't return back to the previous step, or we don't return back to the first step, right? But an analyst, when he sets into the lab, even if 8 months are completed in the year of research, if he is required to start all over from the first step because of some validation issues, he or she should not mind going to step 1. That is what is the key understanding of analytical chemistry. Without the need for going anti-clockwise anytime. I just want to highlight on importance of validation, essential in proving the quality, reliability, and consistency of a chemical being developed. Proper validation of a method provides documented evidence of a method performance and prescribes ongoing measures to ensure quality monitoring for the life of the product. So good laboratory practices are something GLP, which is to be practiced by an analytical scientist, should always have a practice of documentation. Written SOPs are required, and the laboratory should ensure that this SOPs are in place, which will describe the entire process of conducting method validation, so that there is no question of any shortcuts anywhere, which are encouraged. The laboratory should use qualified and calibrated instrumentation. I'm repeating some statements because they are the biggest challenges today for an analyst. There should be very well developed and documented test methods and approved protocols prior to the validation. The protocol is a systematic plan that describes which method performance parameters should be tested, how the parameters will be assessed with its acceptance criteria. Placebos and reference standards are needed to perform the validation experiments. Many a times we complete validation without ensuring that placebos were used and the real reference standard was present when the validation program was undertaken. So it all culminates into something which is the highlight of this presentation. It is the need of the robust methods. So the most common weakness in development and validation of methods is that the methods are not robust. So if robustness is not built into the methods early in the development, then the results are likely to lack efficiency in quality testing and encounter lengthy and complicated validation process. So robustness is achieved by conducting forced degradation studies. So you predict what are the possible impurities which are likely to come along with the API or the drug product, and see that the method is developed in such a way that even if those impurities are formed, they should be simultaneously analyzed along with the compound. Today, you must have seen in good journals, it is only those methods which are stability indicating methods which are accepted for publications. Simple methods are not accepted at all. Gone are those days. So the validation of an analytical method demonstrates the scientific soundness of the measurement of characterization. The validation practice demonstrates that an analytical method measures the correct substance with high level of specificity in the correct amount and in the appropriate range for these samples. Validation should allow the analyst to understand the behavior of the method and to establish the performance limits of the method. So why it is challenging? Why analytical method development is challenging? The problem is the method validations. I've already pointed out to the gaps earlier. Incomplete method validation persists as a significant problem in pharmaceutical development and manufacturing. When not performed properly, it can result in delays in product approval, require a complete do-over of API development, or cause regulatory delays, because a lot of questions are asked by the regulatory authorities which will not have answer with the developed method of analysis, and that could be one of the source for delay of the release. So being aware of the common mistakes, knowing how to avoid them will help ensure analytical methods successfully validated. So prior experience, so this is what I'm trying to highlight here, is the role of the guide, role of the mentor is very important when we allocate projects on analytical method validation for our students, because we are aware with our experience the common mistakes which go into analytical method validation. So what the perspective of FDA, I think all of us are, as teachers, we are all aware of this. So I don't want to spend more time on this slide. I just want to complete my presentation in the next 5 minutes. The FDA provides clear guidance on method validation requirements, which is closely aligned with the ICH. According to the guidance, early in the development of new analytical procedures, the choice of analytical instrumentation and methodology should be selected based on the intended purpose and scope of the analytical method. The parameters evaluated during method development: specificity, linearity, LOD, LOQ, range, accuracy, precision, whether it is FDA or ICH, they talk about these parameters. And the biggest issue is manipulations which affect, the performance of a method that triggers the robustness, that ensures that robustness is not up to the mark. So analytical procedures in the early stages of development are initially developed based on a combination of mechanistic understanding of the basic methodology and prior experience. Once again, it signifies the role of the guide or the mentor in analytical method development. So quality by design is something which is growing these days, where every parameter right from beginning is given due cognizance when method development starts. Many times we ignore the chemistry which goes into the analytical aspect, and this could be one of the reasons for ignorance. So when you go for that cookie cutter approach, you don't take cognizance of individual differences. Then what happens is something that has been done by seniors is something which I'm following now, or something which I have done in my MPharm or PhD project is something which I'm doing for my students after 10 or 20 years. So I'm not changing in tune with times. That is called as the cookie cutter approach. When you follow, then you invite trouble. The method may not be robust. You should know that technology has so developed over a period of time. Even within a few months, technology upgrades itself to such an extent that something which we have not seen 6 months ago in the product can be visualized today. That is the positive side of the technology which has got into the analysis today. So typical culprits in method validation problems, they can be found even before validation studies begin, and the root cause is the lack of thorough understanding of the physico-chemical properties of the molecule. I want all the guides to please note that when somebody decides about analytical method development, ensure that physico-chemical properties of the molecule are properly understood. If not, there could be some problems later on which could be faced with regard to the stability of the molecule, the stability giving up presence of degradants after the product has left the industry, which could ultimately result in recall of the product. So when methods are being developed for a particular drug, ensure that the physico-chemical properties are well understood, and in tandem with that, the methods are developed. Many a times we are not aware that the compound is light sensitive or moisture sensitive, and we would have developed methods only to face problem later on. Only when the physico-chemical nature of the compound is firmly established can an appropriate validation study be designed. Many a times, analytical thesis on analytical techniques doesn't contain any information about the physico-chemical nature of drugs. That means, the design of the method lacks proper understanding of the molecule itself. That is the basic issue. Aside from improper assessment of the physico-chemical properties, there are three common mistakes which occur in the method development. The basic objective of the study itself is ignored. Not thinking ahead to future methods testing, you should be aware that when the product comes out of the market, it's in the public domain. Anybody can take that drug and analyze and unmask the efforts that have gone behind development of the drug in the industry. If robust methods were not applied in the industry, there's no question of that product surviving the shelf life. These are the questions that often come into mind. The purpose for which the method was developed itself will not be known to many students. Whether the method was developed for the release of raw material, is it meant for development with regard to in-process control? Was the method developed for the purpose of releasing the final product, etc., etc.? Unless the objectives are well laid and the analyst knows for which purpose the method is being developed, you cannot expect robustness of the procedure or robustness of the method. So the analytical method can go into analysis of the drug in any one of these conditions, which I have just listed out of my memory. This is what I said. We don't think that once I test and I approve, I cannot be complacent and think that my method will not be subjected to test again by external agencies. So that is where my method fails again if it is not. So the backbone of the issues is basically, the issue is with regard to inadequate efforts that have been put during the optimization process. So inadequate efforts, or the efforts are only to make a thesis, that is the biggest issue what analysts are facing these days in many departments. I would just like to summarize my presentation in one or two slides. The analytical methods that are scientifically sound, well understood, and properly validated with the regulatory pathway in mind are the basis for successful manufacturing and regulatory approval, as well as safe and effective drugs. I think we all accept this. lack of availability of reference standards and impurity standards, extremely low levels of impurities in test samples, low sensitivity of analytical methods that are developed, lack of clear guidelines on acceptable assay variations, etc., could be one of the sources for problems, and that could be taken as challenges. Many of these challenges can be overcome by adapting appropriate sample preparation techniques, sample enrichment techniques, or improvement of the assay range. I think this is where analyst has a role. Analyst can define and redefine the conditions, redefine the procedures, make them complex if required, and ensure that robustness is fed into the method. The most important part is no bias, no manipulation. And if there is no bias and no manipulation, I think that can lead to pathway of analytical method which will be developed to be robust. The moment bias and manipulation comes into mind, it is as good as saying that the method finally will be non-robust. I think I would like to thank here the organizers for giving me a chance to share my thoughts on the challenges which are faced by analysts when methods are being developed in the analytical laboratories, whether in academics or in industries. The first frustration that creeps into our students when they start working in industries, because the treatment what we provide them in the academics, where submission of thesis is the major concern, and when they reach the industry, the kind of treatment they are meted out, because they are not exposed to stringent conditions in the institute laboratories, they really face the reality when they go to industry and they are required to run pillar and post, and then struggle for some time until they get adapted to the needs of the industry, where they are always on the lookout for the most robust methods. I think I should thank the participants for giving me an opportunity to share my thoughts, and perhaps if they are taking up projects on analysis, I think their motto should be only to take up those projects which will eventually lead to development of the robust methods.
Thank you, sir. Just a few questions from any delegates.
Yeah.
So there's a question for you.
Yeah.
Good morning, sir.
Good morning.
So my name is Prarambhi and I am from Kelly College of Pharmacy. So I'm a student, so I want to ask you a question. Like when we follow a particular research paper for analytical method development for the procedure, so now how do we supposed to know what is the method to be like the method what we develop is correct, or can we rely on the research paper which we follow?
What you perhaps will have to do is take up a paper which is peer reviewed by the best possible experts. Go to those papers where they come from very high impact factor journals. I'm talking of them because many of these journals, because I was an editor for some time, from my editorial forum I know that there are some journals where the matter that has been received by them for publication is sent to some identified laboratories where they actually do those methods and see whether those methods are reproducible and good enough to be considered robust. It is done practically, and then when several questions come up, they are addressed by the authors. They are sent to the authors by the editor, and the authors respond giving proper justifications. So what you have to do is when your students develop a method, you have to subject it to extensive criticism, and then if available, take up that method for comparison with what you think is the most robust method. Pick up those methods which have been reported in journals. Compare your methods with the reported methods. But first, you should ensure that the methods that have been reported by your students are bias free. Most of the time, they are biased methods. Biased in the sense they ultimately fit within the acceptance range. But the moment you look at some parameters, you will get a doubt. You just ask them what is the source for the formula, what they have used. The formula will be something which will ensure that they get an answer which will fit within the acceptance range. So to give you a one-line answer, unless you compare your method with the method which is most robust, which is published in high impact factored journals, where they take due precautions before it gets printed, where those reports, those methods are subjected to critical reviews by experts, and if required, subjected to analysis in some laboratories for the purpose of ensuring robustness. Pick up those methods, and then if your methods fall or meet their expectations, then you can consider that it is good enough to be considered as a robust method, which is very robust. Otherwise, no. Unless you compare it with a standard method, you cannot declare that your method is robust.
Thank you, sir.
Thank you so much, sir.
Thank you.
Anybody? Yes, there's something from.
Sir, there's one more question from
Yes, please.
Anand Bandarkar.
Yes, yes.
So the question is, what is the role of industry in developing robust analytic method in terms of institute-industry interaction?
That's a big question. That's a very strong question, in fact. And the industry supports students in terms of educating what is considered as the robust method. They educate the students. In fact, my experience is the students are made to take up, they really are taught, they know what robustness means. Robust is just a ROBUST, a 6-letter word, but the amount, the quantum of work that goes into making of that, the method to fit within that robustness, what is defined as robustness, is so exhaustive that every student who goes to a good industry for a project will come back with a lot of experiences to share with and enjoy sometimes. And then, what is reported by them in their thesis may not totally reflect on what is done by them in the industry, because the industry uses them initially when they are training them. They will use them to develop robust method for something that is required for them, in the sense required for a product which is fast moving currently, or if they have some kind of a regulatory issue with regard to some project, then students are used for that particular purpose. The advantage is student gets to know the in-depth intricacies of what robustness method is. Now, some of them, which whoever who likes to take up those challenges, will definitely go back after their PG studies or PhD studies back into the industry, and then fit in that particular groove. If they don't enjoy, then they are out of it. They just move out, and then they would not like to go back. They will say it is all very monotonous. It is all something which will eventually lead them to be under that collar of a bad boy or a bad girl, because you know that when a product is rejected, it is the analyst's name which takes the bad, and something is passed, nobody cares about it. But when some batch is failing, when regulatory authorities tomorrow come and punch the industry, finally the victim is the one who has developed that method. Because that method is not robust, there were a lot of loopholes, there were a lot of gaps, and that is where the product eventually goes up to that stage and starts failing. So the person who normally takes up these kind of projects, if they are trained in the industry for developing robust procedures, I repeat, robust procedures, then if they would like to continue their career in that, it is fine. But many a times we have seen that they cannot sustain that pressure of developing that robust procedures. They fall wayside and get into some regulatory or some other area because the accountability is not that high as the accountability rests with the testing group. The testing group is the one which is sitting on the hot chair there, because tomorrow if anything good happens, nobody talks to them, but if anything adverse happens, then the falling sword is right on their head. But it's good to take up these kind of projects in association with the industry, because that is one way of learning what is correct and what is not correct.
Thank you, sir. I think we got to conclude the session since we are running short of time. We'll have to start with the next session. Sir, I would express my gratitude and for this your session on in very simple and impactful presentation that we have given, and the strong message also that has been conveyed to the teachers for emphasis on grooming students and to keep abreast with the changing technology. I also also the young teachers have to take in mind about training their students on use of volumetric measures, which is very important at a learning point. At the starting of I think we got to train our mind students first initially, so that they grow up to have their better research attitudes. Thank you, sir, very much on this session for sharing your expertise and experience in impurity analysis. On behalf of KLE College of Pharmacy and AICTE, I extend my heartfelt gratitude for your valuable time. Thank you, sir.
Thank you. Thank you so much. And I have the pleasure to conclude and then thank the management of KLE to give me an opportunity so that I could speak to your delegates. And then perhaps if I have seeded a thought in their mind that it is eventually the ethics which count when it comes to safeguarding the humanity, the health of the human beings, the fellow human beings, then it is from the analytical testing that it should start with when it comes to the industry. So if you are not compromising type, you value the ethics the most, then you are the right person to be considered as an analytical chemist, because it is your performance that is going to say yes or no for the product which is coming out of the industry. So thank you so much. I wish all the participants all the best, and have a pleasant time ahead in this QIP as well as their careers over the next few decades. Thank you so much. Yeah.
This is a very strong message conveyed, sir. As pharmacists, as a professional, where we got to maintain our integrity. Thank you very much, sir.
Thank you so much.
So you can take your leave.
I'll log out. I will log out.
Okay, okay.
Yeah.
We have our next interesting session by Dr. Chandramouli H, which will be, and the speaker is already is in contact, and he's waiting with us. With a very short break to start with the second session. We hope we get you back on time. Chandramouli sir, please a few minutes of time to start the next session.
Sure, we'll be start by 10:35.
Sir, it's 11:30.
Sorry, 11:35.
So we'll just take a few minutes gap.
Okay.
So that we can just close this and start with the next session.
Sure.
Thank you, sir. Participants, please join for the next session on time. Thank you.
Ma'am, can you enable my screen sharing?
Sir, I think we are doing the recording. So it would be a very long session to record both. So we'll just start with another sharing, sir, with you.
Okay.